I'm James Grissom. I'm a postdoctoral fellow at the University of North Carolina at Charlotte, and I'm really excited to talk to you a little bit today about our use of 3D printing and more recently, actually virtual reality, to educate undergraduate students about DNA protein interactions. And also, I would like to say it's actually really exciting that this is more of a zoomed in view of a specific activity that we've been doing with students, but it's actually really heavily influenced and based on one of the cure courses that our keynote speaker, Dr. Sarah Brownell, actually had a huge hand in putting together. So it's really cool to kind of have her here to see that as well. There we go. So first, a little bit of background. So the overall goal of what we were trying to do here was to develop an active learning activity that would help teach undergraduate biology students about the different levels of protein structure and how mutations in the amino acid sequence can cause some defects in a protein's ability to function. So to do so, we used this protein called P53, which is colloquially named the guardian of the genome. Right. So just a little bit of scientific background. Its main function is to transactivate genes that regulate the cell cycle, in the presence of DNA damage. And so I have highlighted here for your convenience. We have in this image on the right our DNA kind of highlighted in purple. And the different monomers of this protein can then interact directly with the DNA. And depending on how much damage there is in the cell, it can either promote cell cycle arrest, or if it gets really, really bad, it can actually induce apoptosis. So we sought to use this protein as an example for teaching students about the different levels of protein structure and how these proteins can interact with DNA. And specifically, when we first started to try this activity, we were taking students that were taking a junior level kind of cell biology courses. So for the activity itself, we actually developed these P53 model kits that I have shown here. So using 3D modeling, we took the crystal structure of the P53 protein as it attached to DNA and 3D printed them for our students. So for the actual activity, we gave the students an initial kind of pre-lab sort of quiz just to gauge how much they already knew about protein structure. And then we gave them a short lecture on the structure and function of P53. And then the actual hands-on part of the activity, we gave each student one of these model kits, and we had them paint them with the different levels of protein structure being different colors. Right. So for example, you could take, beta sheets and paint them red, alpha helices, paint them blue, unstructured regions yellow, the DNA green, so on and so forth. And so, following that activity, we gave them a short kind of qualitative assessment to gauge what they enjoyed most about the activity, what they found difficult, and their thoughts on, how to improve the activity in the future. And then following that kind of qualitative assessment, we gave them kind of a post, almost like a post lab, very short kind of quiz that they took after the activity to gauge, how much they actually learned from this interactive activity. So the main positives that we got back, and the ones we've compiled from the many iterations that we've done over the years now with these students, is that they really enjoy the hands-on aspects, and they found that having the 3D model actually makes it a lot easier to visualize what secondary protein structure looks like, how these structures fit together into tertiary structure, and they can really visualize how this protein P53 can directly interact with DNA. So some of the main drawbacks of this kind of first trial run of the activity, it mostly had to do with the difficulty of the painting. So one, paint's very messy, right? But also, as you can probably tell from the model I have pictured here, to get to the internal sides of the model, even with a really small paintbrush, it's really hard to access those without getting paint everywhere else. Right. And also, as cool as these models are, because it does give students something tangible to work with, we found that they actually break extremely easily. Okay, we have very small connector pieces that keep the P53 kind of attached to DNA. And if you accidentally drop the model, it's liable to kind of snap. So taking this into account, these responses really helped us to kind of reform this activity, especially after COVID hit and the lockdown occurred. We really had to find a way to be able to try and do this virtually. And so that really led us to be able to revamp this activity by shifting to an online program called Paint 3D, which you can download for free off of any computer on the Microsoft Store. And so taking the model that we used to print the the actual physical version of P53, we were able to import that file into Paint 3D and allow students to do this virtually. So this is good for our side of things because we no longer have to mass print 3D models. So just to give you an idea, to print 4 of these models at once, it takes about 7 or 8 hours. And you can imagine if you have a classroom of 24 students, you have to prepare weeks in advance to get all of these models ready for student use. So there's also a ton of benefits for the students. So, theoretically, they don't have to worry about getting paint everywhere. It's much easier to move the model around and zoom, as I kind of have on the 2 videos on the slide here. And it's really easy to access these hard to reach spots. And, because it's virtual, if you mess up, you can just hit Control Z to undo your mistakes. And so, as a result of switching to virtual reality for this activity, which we've been doing in recent years, we found that the students really enjoy it a lot more compared to the previous iterations. So, in the sense in the assessments that we've given them since switching to this virtual version, they really highlighted how easy it is to move the model around, how to paint the internal sides, and what was really important is we could actually have the students really zoom in on where the protein is interacting with the DNA so they can see how the 3D structure of the protein allows it to fit into DNA's major groove. And so this is a lot easier to do virtually than with physical models because virtually you can keep zooming in. If you have a physical model, you can only hold it so close to your face to see it. So there are only 2 main negatives with this virtual reality that we've seen. The first is that it is a little difficult at first to learn how to use the Paint 3D software. The second problem is when we first started doing this virtual version of it, we were having students share computers because at the time we didn't have enough computers for every student, so we were kind of pairing them off, sometimes into groups of 3. And so that did become a huge time sink to allow students to kind of share the computer to paint the models. But with this feedback, we've been working on kind of rewriting our instructions to use Paint 3D so it's a little more easy to follow. And also, we now encourage students when we're doing this activity with students in different courses to bring their laptops to class so they don't have to share resources with each other. And so finally, just a few future directions that we plan on doing. So we're working on creating 3D models of P53 that actually highlight specific mutations that have previously been identified in tumor samples. And so what we can do with our 3D model is actually find where these mutations are and kind of highlight them for the students. So we can use that as a way to also teach them about how mutations in proteins can lead to sometimes disastrous effects. So thank you all again for your time and your attention. I hope that you found this activity a little interesting. I'd like to thank, everybody at University of North Carolina at Charlotte in the biology department who have helped us kind of refine this activity. And again, I'd like to thank our keynote for helping publish that amazing cure study that really inspired this activity. And if you have any questions, I would happily address them.